Welcome to our brief lecture video series for nurses, and today we're going to look at immunoglobulins, or commonly known as antibodies. Okay, so the content that we're going to discuss today is introduction to the various concepts about how antibodies are and what they are and what they do. And then we're going to look at the functionality and then structure, which is very important. Then we are going to finish up with the different classes and characteristics of each class of antibodies. Okay, so just as a brief introduction is just immunoglobulins, which are normally denoted as Ig, also referred to as antibodies. Okay, so and normally they're called antibodies because they were found to be acting against certain bodies or foreign substances, which we'll later on see that these foreign substances are called antigens. So antibodies basically may be defined as proteins. They are proteins in nature, and their main function is to neutralize microbial activity or even foreign substances such as bacteria, viruses, and other foreign substances. So immunoglobulins that usually come out or they are emanated from B lymphocytes. So the B lymphocytes, once they mature and they are activated, they release these immunoglobulins. So substances that can evoke antibody, not anything, even though we've said that antibodies act against certain foreign substances. Not every foreign substance can evoke an immune response or can evoke antibodies to start doing their function. So these substances that actually evoke some activity from an antibody is called an antigen. Okay, so and we will look deeply into what antigens are. So normally, the antibodies or immunoglobulins exist in two forms: those that are bound on the membrane of the B lymphocytes. Okay, so these ones normally act as receptors for antigens. Okay, so there are those that come bound on the B lymphocytes, and there are those that are found in the circulation, in the blood circulation and in the connective tissues. So those are the two main forms that we can find antibodies in our body. So how do they act? They act in two main ways, and that is basically what we have alluded to before, is that antigens antibodies act by doing what we call antigen recognition. They recognize that this is a foreign body, okay? And then after that, we have what we call outcome determination. Okay, it determines what is going to happen. Is it going to be marked for phagocytosis? Is it going to be neutralized directly? Are we going to have complement system coming in? So those are the two main functions. So they recognize and then determines what is going to be the outcome of this foreign body. So and to do this, normally antibodies have two different sections. We just call it moiety, but put it simply, two regions. So we have a region of antigen binding sites. So these are called the Fab, the fraction of antigen binding. That's Fab. So this is it's very variable because it's variable because we have so many different types of antigens. So each antigen will have a different antibody, and the difference comes especially with the difference in the antigen binding site. The rest of the sections of the antibody can be the same, but what is really varied is that antigen binding site. So it's very varied and it's very specific. So yeah, this is a highly variable area which determines the specificity. Okay, the Fab. Then we have the FC region. This one is where now binding of antibodies and antigen interaction occurs. Okay, and it's called the fraction of crystalline. Okay, and the name basically emanates from the crystals that were being formed when these sections are separated or they're broken down. So we have these two sections, the Fab and the fraction of crystalline, which is FC region. So the structure basically, and this is very important, the structure of an immunoglobulin is Y-shaped, and there are a lot of studies that are done to actually determine this kind of shape, to determine that immunoglobulins have this kind of shape actually. So it's not just a fragment of imagination, even though not all of them look like this, but this basically the general structure. So they normally have two chains. Okay, so with two peptide chains. Okay, so there's this chain in the middle. Okay, all through down. This is called the heavy chain, and then we have another pair of chains on the outer section, normally just found on the upper section, and these are called, this one and this one here, they're called light chains. Okay, so but the terminal section of both the light and the heavy, which is found on the antigen binding site for antigen recognition, these are called the variable regions. And I say it because they keep changing depending on which type of antigen we're dealing with. Okay, so it has to be more specific. And then this other section is, the rest of the other sections are called the constant regions. Okay, they normally don't change. So this is a constant for the constant heavy chain. These are constant. This part is a constant light chain. Okay, so all down, this is constant region. So this, as we said, these are effector functions are directed by the terminal constant region. So these ones, it's where we also have the FC. Okay, FC is made up of the constant region and determines what now basically function we're going to do. So these chains, the chains for the heavy chains actually are held together by disulfide bonds. Okay, disulfide means just two sulfur bonds, and that is what holds it together. Okay, this is another schematic representation, and this basically it's looking at, you see there's a variable heavy chain and then there's a constant heavy chain. One, constant heavy chain. Two, not, they're divided into sections. Constant heavy chain. Three, you might even have four depending on which type of immunoglobulin you're having. Okay, so basically you have the heavy chain, the light chain. Okay, and then these are the different regions we're having. Okay, so simple, we have heavy chains, which goes down, and then light chain. Okay, just so that we understand the difference between these two chains, the heavy chains, these are the heavy chains we've talked about. The heavy chains mainly are divided into 5 classes, which each is given a designated Greek name. So we can have gamma, which is IgG, immunoglobulin G. We can have alpha, that is immunoglobulin A. We can have mu, which is immunoglobulin M. We can have delta, which is immunoglobulin D. And we can have epsilon, which is immunoglobulin G. Okay, so by the way, these are structurally and antigenically distinct, meaning they're different in how they look like. And we'll discuss this even more. And you'll notice that actually heavy chains is what now determines the classification of an immunoglobulin. Actually, we call IgG. Okay, we call immunoglobulin G because their heavy chain is made up of gamma chains, gamma heavy chains. Okay, so basically heavy chains are the ones that determine the different classes that we look at. The light chains, we mostly have 2 types. Okay, but you can only have one type in a single pair of immunoglobulin. Okay, so we have kappa, the kappa type of light chains, and we also have lambda. Okay, so you cannot have both at the same time. You only have either. So you can have a kappa light chain in an immunoglobulin, or you can have lambda. Okay, so those are the 2 types. Okay, so now that we have understood that, it's also important we look at functions before now we look at structure. Normally we start with structure and then we go to functions, but in this case I would prefer we look at the general functions. Okay, so in this case what we're talking about is that the main function, even though these are general function, even though each immunoglobulin has a different specific function, which you look at when you're looking at individual immunoglobulins. But generally, their main function is to neutralize, especially bacterial viral particles. Okay, so what we're looking at here is an antibody, and then there's a viral particle which just come from an infected cell, and then it binds. Okay, then it is inactivated directly. So by binding and neutralizing it, okay, which is very important. Also, we have opsonization. Opsonization basically it's like binding to the antigen, and then it becomes marked for phagocytosis. So that is basically what opsonization is. So what happens is that if it's a bacterial cell, we have antibodies which come and bind, and then now the phagocytes know that this one is marked for engulfing and phagocytosis, okay, using the receptors, and then it comes and takes it up, okay. And then we have the complement activation. We have a complement system, and especially the classical pathway. This one, the immunoglobulins actually now start off the process, okay. So they activate the complement system, and specifically the classical, because we have other complement system pathways like alternative pathway and the lectin binding pathways. So, but the classical pathway is normally started by the immunoglobulins. And then activation of cellular elements. And this one is important because sometimes once the immunoglobulins are attached to identify an antigen, then it sends signals for other cellular elements to come and help in fighting the infection. Okay, so the different classes that we have, as we said before, they're based on which heavy chain we have, and they can be placed into 5 major classes. We can have IgG, IgM, IgM, IgA, IgD, IgE. Okay, and this is basically how they look like. And so we'll look at each one of them individually so that actually we make sense of them. Okay. So if we start off with immunoglobulin G, so it's made of the gamma heavy chain, so it's called G, and the structure is a monomer. A monomer, basically from this sense, you'll be hearing this term, a monomer, dimer, a pentamer. So monomer is basically a single structure, a single complete structure, the Y-shaped structure of an antibody occurring alone. Okay, so like this one is a complete Y-shaped structure occurring alone. So this is an IgG structure, so it's a monomer. Okay, so the percentage of serum antibody, actually IgG is 80% most abundant, and obviously it has the biggest role. It is the most abundant type of immunoglobulin, okay, in our serum. Okay, and that's an important fact that we have to know. And because of this, it's predominantly it's the predominant antibody in the secondary response, and constitutes an important defense against bacteria and viruses also. So it's found in blood, in lymph, in the intestines. Okay, and also immunoglobulin G has subclasses: IG1, IgG1, IgG2, IgG3, and 4. So, and we know that it crosses the placenta. And that's an important fact. It crosses the placenta, therefore it can infer immunity from the mother to the baby, okay, or to the fetus. So the main functions that are known for this, they enhance phagocytosis through opsonization, and then they neutralize toxins in viruses directly, and also they offer protection to fetuses and newborns. Okay, so that is just brief overview of it, and you can easily understand IgG is very important. Actually, like they look when you've heard of the issue of serological antibody tests for like COVID, and what they look at is IgG and IgM also. which we're going to look at. The important immunoglobulins that are produced in response to a viral infection. Okay. So yeah, let's look at IgM now. IgM is a very interesting immunoglobulin. So for this one, the heavy chain is mu, and that's why it's immunoglobulin M. The structure is a pentamer. Okay, pentamer meaning five complete Y structures. So you see this is one, another second, third, fourth, and fifth. Okay, and then all of them are normally joined by a disulfide bond called the J. The joining chain. Okay, so with that, so you can see it's a big basically structure. It's a big kind of immunoglobulin. So the location, before that, the percentage in serum is not as high as IgG, but it makes up 5 to 10%. It's normally found in blood, lymph, B cell surfaces, okay, as a monomer. So remember we said you can either find immunoglobulins in two forms. You can find it on the cell surface or the B lymphocyte, or you can find it in the blood or in the serum. So in this case, IgM can also be found in the B cell surface, but in the B cell surface, it occurs as a single Y-shaped IgM. Okay, that is a monomer. But now once it gets to the serum or blood, lymph, so what happens? It becomes a pentamer. Okay, so it is predominantly intravascular, and it is normally involved in the early immune response. So IgM is a bit different from IgG in that IgM is very important for the primary response. So the first time you get an infection, okay, a new infection, then the first responder, okay, or the predominant responder is an IgM. Okay, obviously later on you'll get an IgG. Now, but when you get a secondary infection, okay, then the biggest response is by an IgG. Okay, so IgM is not as much in a secondary response. Also, the placenta transfer, it does not get transferred by the placenta. So the functions for immunoglobulin M, it's the first antibody produced during an infection, and it's effective against microbes, obviously, and it's a major class that is also involved in agglutination. Very important. Okay, agglutinating antigens. So this is basically what is also important in blood groups. Remember, for example, ABO blood group system is that you have an antibody against and you have an antigen on the surface. So what helps in knowing is which type of blood group you are is the agglutination. Okay, so also in autoimmune diseases, and autoimmune diseases, it's our own antibodies acting against our own cells or tissues. Okay, that leads also to some sort of clamping together. Okay, autoimmune disease. So IgM is very important for this kind of activities. IgA, very important one also. It's of the alpha heavy chain, so it's IgA, and the structure is interesting. Okay, it occurs as a monomer in the serum, but as a dimer in a secretory IgA. Okay, that is in secretions. Okay, it's important also that IgA normally is important for in the secretion bits, like in our saliva, in our tears. Okay, think of our secretions that we have: saliva, tears, the secretions in the intestines, milk produced by mothers. Okay, those are secretions. In secretions, IgA occurs as a dimer, but in the serum, okay, that is in blood and around lymph, it occurs basically as a monomer. Okay, so it's important to know that it's also joined by a J chain. We talked about the J chain in the pentamer IgM. Okay, so the percentage is around 10 to 15. Location of this immunoglobulins, mostly secretion, that is tear, saliva, intestines, and milk, and also in blood and lymph. So it is taken up by the epithelial cells, and the secretory piece is added. And this ability of this IgA to be taken by the epithelial cells makes it a bit resistant to enzymatic degradation. Remember, most of our secretion have some sort of enzymatic secretions that actually break down foreign bodies, let me put it that way, or microbes. So for example, saliva or tears, okay, they have some protective or immunological activities. So what helps the immunoglobulins from being broken down or lysed by these enzymes, just they're taken up by epithelial cells. It does not get transferred. Okay, so this is another second immunoglobulin that also helps kids who are breastfeeding, because now they're given the IgA through the breast milk. And as you can see, we said it's a dimer in secretion. There's one and another one here, and the J chain here. So the function, it offers localized protection of mucosal surfaces. So remember, most mucosal surfaces are covered by secretions. So we have tears, saliva, secretions of the intestines. Okay, milk now going to the intestines of the baby. Okay, so through that we have provision of immunity to infants' digestive tract also. Okay, so J is very important also. So IgD is of the delta heavy chain, and the structure is a monomer, simple monomer. Okay, however, IgD is not very abundant in the serum. So the percentage serum antibody levels are 0.2%, and the location it's found on the B cell surface. Okay, if you remember, another immunoglobulin that is found in the B cell surface is IgM. Okay, so but it's also found in blood and lymph. So it is associated with the surface of the resting B cell. Okay, the one that has not been activated. Okay, to become plasma cells, so that it produces more immunoglobulins. So it's normally together with IgM. Okay, but the only immunoglobulins that you'll find resting on the cell surface of on the cell surface of a B cell, which is resting and not activated, that one is either IgM or IgD. Okay, so and I think I have a picture here. You can see this is a B lymphocyte that has not been activated, and then you have IgM and IgD. Okay, these are the normal. Then later on we have now class changes, and we have other productions once it is activated. So it is not transferred via the placenta, and the functions, it's normally found on the B cell surface, and it initiates immune responses. It may be important in B cell antibody binding and subsequent differentiation. So when you have an antigen coming and then attaches, it activates now the B cell to actually now differentiate to plasma cells, basically to produce now specific immunoglobulins for that antigen. Finally is we have immunoglobulin E, and it's of the epsilon heavy chain. Okay, and as you can see, it has a longer FC region. So the structure is a monomer, simple monomer again, like IgD, sorry. The percentage serum antibody levels is minute, it's 0.002%. Location, it is bound to mast cells and basophils through the body and also the blood. For that reason, it's also important for allergic responses. So IgE mediates the immediate hypersensitivity, because if it's found on the mast cells and you have an allergen, which is basically an antigen or a foreign body, and attaches to it, it activates the mast cells, okay, through its FC regions. So it binds, and we know mast cells have histamines or mediators that are inside, and once they're released, they cause that reaction that we call an allergy or a hypersensitivity reaction. They do not cross the placenta barrier, okay? But apart from even allergic response, IgE is also important for lysis of worms. So they also attach to worms, and they cause lysis of the worms. Okay, so helminthic infections will also indicate a rise in IgE levels. Okay. Good. So basically what we've talked about, the different types that we've looked at is IgG. Okay, it's it's a monomer. It's the highest level. It has the most abundant. It's important for opsonization, neutralization. It has the four different classes. IgM, it's a pentamer in serum, but on the cell surface of a B cell, it is a monomer. So it is produced fast upon antigen invasion, increasingly transiently. Okay. We said IgM is very good. It's the predominant one for primary primary response. IgG is very important for secondary response. IgA, it's a dimer in the secretions, and it's a monomer, occurs also as a monomer in the serum. Okay, and it expresses mucosal tissues, and it forms a dimer after secretions. IgD, it's a monomer, and it's written here non-function, but we know it's found on the cell surfaces, and it can also initiate the activation of a B cell into plasma cells for differentiation. And then we know IgE, it's involved in allergy. It's also a monomer, and also it is involved in helminthic infections. Okay, so this can show just a brief distribution in the body. IgA in secretions, and lining the mucosal surfaces. Okay, IgE, IgM, they're mostly found in serum. Okay, and other tissues of the body. So thank you very much. If you enjoyed it and liked it, just feel free to actually like and subscribe to our YouTube channel for other more videos. Thank you very much.